Yeah, the small orange flags are our flags, and we basically, put the pitfall trap into this hole, make sure it's the right site. This is F, I want this that way. Plop it in. They're having their normal life, just walking around throughout the plots, and then as soon as they come over the trap, they just fall right in, and they're basically toast after that.
The focal plant is called Kincaid's Lupin. It's got those nice characteristic lupin leaves. It's already set seed, so here are the little seed pods. If you were out here a few weeks ago, you would have noticed nice purple flowers. And so this is the host plant of this endangered butterfly species called Fender's Blue.
The butterfly was discovered in the 1920s, and then around about 1930, they stopped finding specimens. So there had been specimens in museums, and then they thought it was gone until they found it in 1989.
This butterfly lives nowhere except for in the Willamette Valley. And most of the habitats where this plant can exist have been converted to agricultural use. So there's only a really, really small fraction of the remaining habitat left. I mean, you could put all of the remaining suitable habitat in the entire Willamette Valley together in one place. I don't know if it would be as large as the Linfield campus.
The ants will help protect the butterfly from predators like the beetles, because the caterpillar has a little, I don't know what to call it.
A little gland?
A little gland that produces sugar for the ant to eat. And so the ants will protect it because of that.
So what we have are 20 by 20 meter plots. These white and pink flags mark the corners. We're working with a group from WSU Vancouver. Cheryl Schultz and her graduate students are looking at the plant community in each of these plots, as well as the populations of an endangered butterfly species in these plots. And what we're doing is measuring how these different treatments of the different plots affect things other than the plants and the butterflies. The grass is invasive. It's actually, this grass here is tall oak grass, and I believe it's native to Europe.
Oh, yellow. No, that's a flower. Yellow flower. False alarm.
You can imagine how hard it is to find our flags when the grass gets this tall, right?
What I want to go into is pesticides or GMOs, biochemistry. And so this herbicide that we're using is actually fairly interesting.
Professor Tilburg is my advisor, and I was just talking about things I need to do, to get ready for med school and things like that. And he was telling me one of the things I was lacking was research, because I haven't really had time to do research with my schedule.
Med schools like to see that you're participating in research, if only because you learn more about how research is conducted, and just in case you wanted to be in medical research. A little swirl, mix it all up. And then I will separate things into little piles based on what they are. Now what I'm gonna do is I'm gonna make labels and start putting them into these little tubes.
I said Formica 14 because there are 14 ants in there, and they're of the genus Formica. On the front here I wrote the date that the tube is from. So this collection tube was put out on 7 June 2017. And this is from plot 16, trap E.